second place team. My name is Horizon. My background, I majored in statistics and computer science, and I got BS degree in this year. I have no domain knowledge in biology, and this is my first time modeling RNA sequences. But I have participated in various competitions held on Kaggle so far, so it was very interesting and challenging. These are some key points from my solution. I adopted the sketch former architecture, which I got familiar after I finished the ASL competition, previous ASL competition held on Kaggle, and I used GRU head on top of the sketch former encoder. And I used shallow, simple 2D convolutional neural network for the BPP matrix, and I added it as a bias to the attention matrix. Libby positional encoding was used for robust generalization for longer sequences, and I used weighted loss with the value signal to noise and trained the model with longer epochs. And I choose additional features for minor score improvements. These are the features. I use some features found useful in the Open Vaccine Challenge to have fast initial conversions. This includes cap-R loop type and eternal forward brackets and predicted loop type with eternal forward. And this predicted loop type was calculated from BPRNA package. And some BPP features was found a little bit useful, but these features were only marginally helped. So I think these should be removed in the future for the simplicity. This is the overall architecture of my model. So there is a low RNA sequence and some additional features, and they embedded into the 192 dimensional vector. And in the sketch former encoder, they have one convolutional neural network block and multi-head self-attention block. So in the multi-head attention, we used BPP matrix added to attention, added as attention bias. And for the BPP matrix, I used this shallow 2D convolutional neural network. I used sketch former as an encoder with some minor modifications. Sketch former showed strong performance early in training and consistently showed faster conversions. And any comp transformer hybrid architecture with the idea of interleaving 1D convolutional network block between the transformer block also gave similar effect. But among those, I found that sketch former was the most efficient. I choose these parameters, and the total parameters for the single model is about 9 million, and its size is about 35 megabytes. And how I selected these parameters is very just straightforward. So if you add, if you use bigger model, you will end up overfitting to the noise in the training data set. So I will talk about it later, but I spend a lot of time to train larger model, because if you know how to regularize the model correctly, you should get a better result with the larger models. But I think the most important part of the most important technique for the regularization is the augmentation, but I could not came up with proper regularization augmentation method for this competition, so I ended up just stuck to these parameters. Alibi positioner encoding, I used it because it claimed to generalize better over long sequences than other relative positioner encoding methods. I used BPP metrics as a attention bias, and it was and I applied it by multiplying some predefined per head values, and it gave some significant improvement. But I felt that it was suboptimal, and I think there should be some better ways to handle the BPP metrics. And I eventually added this 2D convolutional network on top of the BPP metrics, and it was proved very helpful. But I used the 2D convolution. I maintained the 2D convolutional neural network as simple as possible, because when you use this 2D CNN network on top of the BPP metrics, you will end up with very long training time. So it will be very inefficient. So I used very shallow version of it, and the output of this network is shared across our transformer block layers. And this is the overview of how attention mechanism works. So there is a typical attention score from the Korean key matrix multiplication, and we add the fixed value from the Alibi positional encoding, and we additionally add this BPP bias calculated from the 2D convolutional neural network. And this table is from the Alibi positional encoding paper. And as you can see, the absolute positional encoding should be avoided. So I think probably this is one of the reasons why there was a big shakeup, except the top positions. And Alibi positional encoding claims good generalization over long sequences, among other relative positional encodings. This is the architecture of the 2D combinational network applied after applied on the BPP metrics, and it is very simple. This is the training configuration. It's nothing special about it, but I used weighted loss. I didn't use weighted sampling, but weighted loss with this formula. And the single model training takes around 30 hours with single RTX 49D GPU. The result from the training configuration is as follows. I got 0.129 on 5-port cross-validation, and 0.140 on public, and 0.142 on private leaderboard with a single model. For the submission, I trained the model with the whole training data set. And after ensembling on differences, I got my final score of 0.135 on public and 0.140 on private leaderboard. Here are some ideas I tried and my final thoughts about those ideas. At first, I was motivated to participate in this competition because I thought it would be really cool if I can make self-supervised method running work without using any additional features. But initial trials with some well-known data to bag or BERT-like SSR methods was they showed inconsistent improvements. And due to the additional training time required, I did not consider this further. However, I believe there is still huge potential in this idea, and I saw some other teams made it work. Training larger model, I spent some time on it, but every attempt to train larger models, for example, dimension over 512 with some proper regularizations were unsuccessful. Augmentations, I tried augmentation methods. Most of augmentation methods I tried had no effect, and I had a hard time trying to make some proper augmentation method because I have no background knowledge in this biology and RNA area. And pseudo labels, I tried pseudo label and I thought it might help in a leaderboard scores, but it didn't improve my cross-validation score, so I didn't use it for safety and avoid any additional training time. However, after seeing the correlation between the public and private leaderboard, I think it might have been slightly beneficial.